Hello, everybody. Thank you so much for joining us today. I think we've got a couple of still petering in online, but we might get the opportunity to get started. So much interest in today's Meet the Researchers. It's been phenomenal. So for those of you who aren't aware, my name is Kamara Lawrence and I'm the Director of Advancement at UQ's Institute for Molecular Bioscience. And before we go too much further, I would like to give an acknowledgement of country. So I acknowledge the traditional owners and their custodianship of the lands on which we meet today, or we join in from a virtual space. I pay respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. So, for those of you who haven't joined us for the Meet the Researchers before, this is the first one that we're doing for 2022. And we've been a little bit cheeky about the fact that Valentine's Day is just around the corner and themed it accordingly. And also I would like to highlight that today is actually UN's International Day for Women and Girls in Science. So therefore, the theme has combined and we've got three wonderful women who are going to talk about their passion for all things cardiovascular and their love of cardiovasculature. But a little bit on IMB. We're actually now in our 22nd year. We were the first recipient of what was the BD government's Smart State Initiative. So we had an investment from them that was coupled with the first investment from Atlantic Philanthropies into Queensland, and was really a waterfall moment that then changed the landscape for medical and life sciences research within Queensland. So we have got just over 500 staff here at the Institute, and that's a combination of our incredible workforce of researchers, students, our professional staff. And we really are a melting pot here. We've probably got the better part of 40 countries represented across the faces at the Institute. We have a real strong focus on inflammation, infectious diseases, and as attributed to today, cardiac and vascular biology. And we're doing some great work in regards to things like cancer, pain, chronic disease, and there's a real cutting edge crew in this building doing work around genomics and genetics. So we've got some of the best facilities in the field here at the Institute, and usually the style of the Meet the Researchers is actually in person, but given what's happening in the broader community at the moment, we're doing this wholly in a webinar format. And as part of the Meet the Researchers, people get the opportunity to come in and meet these incredible researchers firsthand and then actually see some of the tech that really drives the discovery that they're able to realise. We are Australia's #1 research institute, which is quite intriguing because we also seem to be one of Australia's best kept secrets. So after today, we challenge you to get out and share that with the community more at large. So the Meet the Researchers was developed as an opportunity to demystify and unmask some of the incredible research that's happening in this building. And I think we've achieved that over time. And so now I would like to move forward and share with you the real stars of today. And first of all, we're going to hear from Dr. Emma Gordon. So Emma joined the Institute here in 2017. She's currently the National Heart Foundation Future Leader Fellow. And her lab is really quite passionate about understanding how blood vessels grow and function. Their whole intent is to develop new treatments for a range of disorders such as cancer, atherosclerosis, diabetes, and COVID-19. And without too much further ado, I'll hand over to Emma to impress you with what their team is able to achieve.
All right. Thanks, Kamara. I'm just going to assume that this is going to run smoothly. So just, Kamara, just yell out if there's any issues with screen sharing, etc. And I'll get this on the road. Right. Okay. It's just loading up. Okay, perfect. So thanks very much for the introduction, Kamira, and thanks for everybody for joining today. It's always kind of fun for us as researchers to do these Meet the Researchers kind of setups, because we get to kind of engage with the public a little bit more than we normally would. So I'm very excited to kind of hear your questions at the end of the session as well. So today I'm really just going to be talking about some of the more recent work that we've been doing in my lab, which is really trying to understand how blood vessels malfunction in COVID-19 disease. Perfect. Okay, so because I'm the first speaker talking in this kind of heart of discovery session, I thought I'd just give a very kind of brief introduction about the cardiovascular system as a whole. So of course it's comprised of the heart, but there's also other critical components of the cardiovascular system that we all kind of need to work in harmony for it to work kind of properly and for our body to work normally. So obviously we have the heart, and we also have the blood vessels and then the lymphatic vessels as well. And I kind of like to think of the cardiovascular system a little bit like a plumbing system. So first of all, you've got the heart, which is kind of like the pump, which is pumping the blood around the body. You've got the blood vessels, which are kind of like the pipes, which transport that oxygenated blood to all of the tissues of our body. And then you've got the lymphatic vessels, which I like to think of a bit like the drains. So if you have like inflammation in your arm, you quite often get quite a lot of swelling. So in order for that swelling to go down, you need to be able to drain that fluid back to the bloodstream. And that's really the role of the lymphatic vessels. So while all of these components of the cardiovascular system are really kind of critical for our normal everyday life, in my lab, we're really interested in the blood vessels. And as I'm sure you already know, the blood vessel network is kind of comprised of these large arteries, which transport that oxygenated blood to our tissues. There's the veins, which transport that deoxygenated blood back to the heart, where it can then, and then you've also got the capillaries, which are the small vessels, which kind of integrate into all the tissues of our body. And we really need the correct specification of these arteries, veins, and capillaries to have a functional blood vascular network. And of course, as I'm sure you know, blood vessels are hugely important during normal life and also in disease progression. And if you look at this kind of, if you look at all the vessels in our body, you can see that they're everywhere and that they're numerous, because they really kind of integrate into all of our tissues, providing them with that oxygen that they need to function properly. So that's why if something goes wrong with the blood vascular system, it can cause such a huge range of diseases, because really vessels are in every organ of our body. So if they go wrong, they can cause problems in every organ of the body. And just as a little bit of trivia, I like to usually throw this in when I'm giving seminars. If you took all of the blood vessels out of your body and kind of lined them up one by one after one another, they would go around the world 2.5 times. So you have a lot of blood vessels in your body, and they're really critically important to function properly. So why am I interested in COVID-19 as a vascular biologist? And I'm just going to kind of briefly touch on the COVID-19 pandemic just as a whole. Even though everybody knows what it is, everyone's probably tired of hearing about it. It's kind of what the second point is meant to kind of demonstrate. You get fatigue when you hear about COVID all the time on the news. We're all tired of it. And you also get fatigue when you get the virus. So it's kind of a double whammy of fatigue. And COVID-19 is caused, of course, by the infection of our body via the SARS-CoV-2 coronavirus. So I'm not going to talk any more about epidemiology or kind of, you know, how COVID-19 has progressed. I'm going to really kind of focus it into the blood vessel system straight away. And that's because really since the beginning of the pandemic, it's really been thought and hypothesized pretty strongly that there is a component to the heart and the blood vessels that goes wrong when we get severe disease. And if you just look down here at this little kind of pie chart, of things that actually cause death when you have severe COVID-19, you can see that there is quite a large cardiovascular component. So, you know, around 20% of patients will die from unexpected cardiac arrest, so heart attacks, and that happens quite often in younger patients as well. They'll quite often get pulmonary embolism, so that's a blood clot in their lungs. They'll quite often die from stroke, and Anna's going to talk a little bit more about stroke and what causes it in the next talk, but stroke is essentially a bleed in the brain. And quite often they'll get a huge range of diseases that are associated with multi-organ failure and the vasculature, such as just uncontrolled hemorrhage, so leakage in our, in our vessels in other organs of the body, sepsis, which is infection of the blood, and ischemia, which is really where you have not enough blood going into your organ, so the organ will die. So we know that blood vessels are really important in COVID-19, and that's why people who are really not virologists really became interested in this virus and what it was doing to the vessels. So just, I'm sure everybody knows this, probably quite a few people on this call actually have had COVID-19, so you already know, but essentially SARS-CoV-2 virus enters our body through the respiratory tract, so that's why we wear masks to try and prevent it. It'll go in and it'll colonize in our lungs. And most common symptoms of kind of mild COVID-19 are fever, dry cough and tiredness. But when it gets a little bit more severe, you start to get symptoms such as difficulty in breathing, shortness of breath, chest pain or pressure, and loss of speech or movement. And when you do get these kind of serious COVID-19 disease and you get put into hospital, up to 30% of those hospitalized patients will develop severe lung disease, which is associated with progressive lung damage. So we were really interested in trying to understand how this lung damage may be associated with the vasculature. So just to kind of go into this, starting off at kind of the basics of how the lung works, because we need to understand how the lung works to understand how it goes wrong. So essentially you have these lung airways, so the alveolar space they're known as. So this is essentially when you breathe in, these kind of sacs will fill with air, and when you breathe out, they will contract again. And this lung airway space is lined by a bunch of cells known as epithelial cells. And then really right next to, so really sitting right next to these kind of air sacs, these air spaces, we have blood vessels. And blood vessels, they're not just a tube, with no cells in them. They're actually lined by cells on the inner layer of the vessel wall. And these cells that line the blood vessel wall are known as endothelial cells. So I'm going to come back and talk kind of quite a lot throughout this just quite short seminar, just kind of referring to epithelial cells. So just remember, those are the ones that line that airspace, and then endothelial cells, which are the ones that line the blood vessels. So what happens when we are exposed to SARS-CoV-2? So we breathe it in, it goes into those kind of air sacs, and it fills these air sacs and starts to really infect the cells that are in there. Okay, so then what happens? So once these cells have been infected, this will cause inflammation. And this type of inflammation that occurs in COVID-19 is known as a cytokine storm, which is just kind of visualized here by these little kind of tornado things. And these little tornado cytokine storms can occur both in the airway space and also in the blood vessel. So this inflammation or direct infection of the blood vessels leads to breakdown of the barrier of the vessels, and then therefore blood will kind of leak out. So it's not being held in the vessel anymore, the vessel breaks and the blood leaks out. And this leakage will really leak into that airway space, and you get basically a buildup of fluid in your lungs. In addition to that, you also get blood clots within the blood vessel of the lung, and these can cause death. So it's both the leakage of fluid and also the blood clotting that's really causing problems in our lungs in COVID-19. So when we started to really think about, how these vascular problems were occurring, we really had to start at the beginning, because we didn't know where this virus was actually infecting. So is it infecting the cells that line the blood vessel wall? So is it lining those, is it infecting those endothelial cells that are on the inside of the vessel wall? Or is it infecting these epithelial cells that really kind of line that air sac? And there's also a third possibility, which I'm not going to talk too much about today, is to see whether the virus can also infect these immune cells, which can also drive inflammation. So we wanted to know, essentially, is this virus directly infecting the blood vessels, or is it infecting adjacent cells, which are then causing inflammation? And then it's that inflammation that really causes the problem within the vasculature. Okay, so I'm not going to show you too much data, just kind of a few snippets. We published this paper at the end of last year. So essentially, the first thing that we did was to look in samples from lungs of patients that had actually died from COVID-19. And we wanted to see if we could see, first of all, if we could see virus in these lungs. And then second of all, can we actually see virus in the blood vessels of these patients that have died? So we have 4 different samples here, so from 4 different patients, so 1, 2, 3, 4. And when we look to see if we can actually observe virus, and the virus here we've just visualized with some red staining. So where there's red, that means there's virus in those cells. And you can see that from these 4 different patients, even if they've died from COVID-19, they don't always have virus in their lungs. So only half of them actually had active virus in their lungs. So these 2 over here, you can see there's really no staining. So just because you're dying of COVID-19 doesn't mean you have a large lung burden. So when we look in a little bit closer in these 2 positive samples, which did have active virus, to see if that virus was going into the blood vessels, and we were able to kind of visualize where the blood vessels are just really by their morphology, by their kind of shape, and the fact that they have red blood cells within them. So when we look to see where those blood vessels are, and if we're able to detect virus in that region, we were not. So there was really no virus in the vessels of lungs from these deceased patients in our samples that we have looked at. So then we wanted to know, okay, can the virus enter these vascular endothelial cells? But to do that, we need to understand how the virus actually gets into the endothelial cells. So I'll go over this fairly briefly, but essentially, when you have this SARS-CoV-2 virus, it has what's known as a spike protein, which I'm sure you've heard of if you've been watching the news at all. And it's quite often this spike that will mutate in the different variants to make the virus bind to cells more efficiently. So this spike will bind to the host cells, and it does this through host cell receptors, which are expressed on the host cell membrane. And once the spike has bound to these receptors, and these receptors are ACE2 and neuropilin-1, the spike needs to be cleaved, and this happens through this protease known as temporis-2. And once basically the spike is bound, the spike has been cleaved, this basically leads to fusion of the viral membrane and the host cell membrane that allows the virus to enter. So we wanted to see, do these vascular cells actually express these receptors and this protease, which will allow for endothelial cell infection. So they do express the receptors. So first of all, we looked for ACE2, which is that kind of primary spike binding receptor. And we have a negative control just over here on the left, and then a positive control over here on the right. And this receptor, we've done some analysis, and you can see the receptor in green. And then just the cells kind of are in pink and blue. And you can see that when we have endothelial cells, which are isolated either from veins or from the capillaries of the lungs, that we do see some ACE2 expression, but it is certainly fairly low, and it is lower than epithelial cells. And we also use some other techniques to look at receptor expression within endothelial cells versus epithelial cells. And you can see that while they express them at low levels, they express ACE2, neuropilin-1, and that protease that allows for fusion of the virus and the host cell membranes. So it seems that they do have the capacity for infection, but are they infected is the question. So we essentially added virus either to endothelial cells that have been grown on the top of a membrane, which will model infection when the virus is in the blood, or we added virus when endothelial cells were grown on the bottom of a membrane, which would really model what would happen if vascular endothelial cells come in contact with virus from the airway. And then we looked to see if they could be infected. And quite clearly, when we looked for viral infection, and here the virus is visualized in green, you can see that when we add virus either apically or basolaterally, we did not observe any viral infection in vascular endothelial cells. And this was really in contrast to our epithelial cells, where we saw very robust infection. And then these were some beautiful experiments that were done by our next speaker, Anne Lagendijk, and she's going to go into this model in a little bit more detail, I think. So essentially, when a blood vessel is in the body, it's not just as like a flat layer of cells, it's actually as a tube. So we grew little tubes of blood vessels, which you can just see over here, and then we added virus to this kind of flow, so kind of where the blood would be, to see if these endothelial cells would be infected if we have like a tube of a blood vessel. And quite clearly, again, when we stain for virus, you can see that we do not see any virus in the blood vessel. However, we do see robust infection of an epithelial cell tube. And this is just these images flipped on their side. So you can see kind of, it's like a little half moon. This is half of the tube. There is no green when we add virus to blood vessels, but there is green when we add virus to epithelial cells. And then finally, we've also used a model of the lung where we grew epithelial cells on the top, which is similar to what we observe kind of in the body, where these epithelial cells line the sac. And then we have endothelial cells on the bottom, which is similar to what we see when the blood vessel is kind of in direct contact with these airways. And then we added virus to the top to model what would happen if the virus came into the airway and had contact with these epithelial cells. And if you just focus your attention over to this side, you can see that when we add virus, we see a robust infection of our epithelial cells, which is just shown here in black and white and down here in pink, but we do not see any infection whatsoever of our endothelial cells. So this is really suggesting that there is no infection of the blood vessel. It's all we're only getting infection in that epithelial cell layer. And interestingly, even though our endothelial cells from the blood vessels were not being infected, they were actually starting to become inflamed, which we can just see here in green. And when we look at whether we were actually getting any cytokines being secreted from these cells, we did see that both epithelial cells and endothelial cells were starting to secrete cytokines. So that's really kind of starting that inflammatory response. Okay, so what have I told you about? I'm just going to wrap up now fairly quickly. So essentially, we've shown that vessel dysfunction in COVID-19 is driven by viral infection of adjacent epithelial cells in the airway, and that's driving inflammation. And when we get inflammation, this occurs both in the epithelial cells and in the blood vessel itself. When we have this inflammation, it leads to a cytokine storm beginning, both within the airway and also in the vessel. And this cytokine storm, so it's the inflammation, it's not the infection of the vessel that's driving the vessel to leak and also driving the formation of those blood clots. So now we really understand how blood vessels are malfunctioning in COVID-19. We're really working to see if we can actually stop this from happening. So can we actually stop this inflammatory response to really try and stop this vascular malfunction and this vascular leak and these blood clots? But of course, to be able to do this, we need to understand how this inflammation occurs, what's driving this inflammation, and really what's in this cytokine storm. Because obviously we need to understand the fundamental biology behind disease before we can actually figure out how we can treat these diseases. And with that, I'm just going to wrap up. And I thought I'd just finish off with this slide because it is International Day for Girls and Women in Science, because this project was really done by a powerhouse team of women. So this was driven by my postdoc, Lillian Schimmel, and it was a really fabulous collaboration with immunologist Larissa Labson, virologist Kirsty Short, and immunologist Kate Schroeder. And thank you very much for your attention.
Thank you very much for that, Emma. That was absolutely incredible. And I must say, I do have a little bit of Christmas envy seeing those beautiful images from the end of last year. It looks like as a collective, you got up to a great time.
Yeah, a Christmas party, yes.
Excellent. I will just share with everybody for their confidence. After each of our speakers have had the chance to give you their presentation today, we will have a Q&A. So I've got questions buzzing already based on all that beautiful information shared with us from Emma. So in the meantime, please, I'm pointing down because your Q&A button should be at the bottom. Please feel free to put your questions in there and we'll do our best to cover off on each of them during the Q&A session that we come to shortly. So thank you, Emma. And you've already alluded to your fabulous collaborator. So without much further ado, I'd like to introduce you to Dr. Anne Lagendijk. So Anne is a truly passionate development biologist, and she came to work at the IMB in 2012. She has a passion for understanding the development of cardiac valves. And there are a couple of things that Emma said before, such as we grow these tubes, but I think that diminished just how truly cutting edge some of the work that is happening, that is being driven by Anna and her team to be able to deliver some of these critical insights. I also, I affectionately call her the fish whisperer. She's tapped into the power of the zebrafish to understand how we can build and maintain a healthy vasculature. And so with that, I'll hand over the duties to moving the slides to Anna.
Hi, everyone. I hope this works. Thank you, Kamara, for the introduction. Thanks to Emma for also a fabulous introduction on the cardiovascular system and how all the aspects, including the heart and the blood vasculature, are so important for this system to do its functions. So I will immediately follow up on that by saying that my lab is really interested as well in blood vessels, and in particularly here this story today. We're looking at blood vessel integrity and how loss of this integrity can lead to hemorrhagic stroke. And with that also, I would just like to draw your attention that at the end of this month, we will have Rare Disease Day. And this day is really meant to raise awareness to many rare diseases out there that don't really get the attention that I think they deserve. And one of these rare diseases I'll be talking about today is called cerebral cavernous malformation. So what is a hemorrhagic stroke? So really, basically, this is a rupture of a blood vessel in the brain. And because that vessel ruptures, what you get is a lot of loss of blood supply locally, and this can lead to death of the brain tissue that's sitting around that vessel. In Australia, we've got roughly 380,000 strokes per year. And of this, 13% is hemorrhagic, so of the bleeding type, while the majority really is through occlusion of the vessel, and occlusion obviously can also lead to oxygen or blood supply loss. And but still, even though this is the minority, these are often very deleterious and also are considered to be more deadly. So the model systems we use in my lab to investigate and study stroke are, as Kamara alluded to, I am the fish whisperer, which is a nice kind of new name for me. I like it. So I've been working with zebrafish for a long time now. It's a beautiful model to study the formation of the heart and the blood vessels live, because we can image all these structures in living animals because they're transparent. And really, we use this as our workhorse for gene discovery. And this is an example here where we've got a zebrafish mutant. And as you can see, where I've written a stroke model, this is one of the genes that we're working on, that when you're lacking this obviously leads to breaking of those blood vessels in the brain. And these fish develop really prominent strokes. But today I'll be talking about another model system that we use in the lab, and Emma also alluded to this, and this is the 3D human blood vessels. I really like this model because we can study human endothelial cells in their normal configuration, and also it's a really nice model to change that environment of the blood vessel into something that's more similar to the environment that we find in our bodies. And I'll go into that in more detail later. So what really prevents rupture of vessels? And one of the main things that prevents this is cell-to-cell adhesion. So this is a dorsal aorta here of a zebrafish. And as Emma said, this vessel is made up out of endothelial cells. And the endothelial cells are all linked to one another by this cell-cell adhesion. And this is all these kind of black lines that you see in this image here is the proteins that are keeping these cells together. So you can imagine that when there's a defect there and you've got not enough of that adhesion structures, then obviously that can lead to like local fragile regions of the vasculature because the cells are not connecting to one another properly. And then when, in addition to that, you're taking into account that this vessel is constantly experiencing the pressure of blood flow, then when you've got these fragile regions, that can really push and cause it to further rupture. So going back to cerebral cavernous malformation, this disease occurs mainly in the brain and spinal cord. And really what happens is you get these really fragile dilated regions of vasculature that are referred to as cavernomas. These can occur sporadically, but they can also be a consequence of genetic diseases. So they can be familial, and also they can be a result of radiation therapy. So quite often we find these cavernomas also in patients that have had brain tumors and then have had radiation, and then afterwards they can develop these cavernomas in the brain. The familial form out of these is the most severe, and we know very well that they're caused by mutations in either CCM1, 2, or 3 genes. And when you look at the cells that are in these vascular regions, it's very well characterized that we find this reduced cell-cell adhesion. This then leads to a loss of integrity and then also this dilation. And then obviously having all of that happening, then that can be, those regions are very prone to leakage. So they can lead to hemorrhagic stroke. So why, so the question we're really interested in the lab is why do these CCMs develop specifically in the brain? Because we know from patients, the familial patients, that they've got these CCM mutations, and obviously they carry that throughout their bodies. Still, they only develop these really fragile regions of vessels in the brain. So what is so special about the brain? And we think this has to do with the environment that these vessels live in, because the brain tissue is quite unique. And we think that maybe this unique environment can induce this CCM formation. So how then is the brain so unique? It's one of the most softest tissues we find in our body. And the reason for this is that really the major component that we find there is hyaluronic acid. And hyaluronic acid not only provides that soft structure, but also it can alter blood vessel behavior. And it does this by breaking it up into different sizes. And each size of HA can have very different effects on the cells. And we know this also from some of our previous work, where we were studying a zebrafish mutant that did not have certain like small fragments of HA. And these fish just completely failed to form the new vasculature that we normally find in wild type fish as they develop. So knowing that, we were really wondering, is the HA content perhaps altered in CCM disease? And because the vessels in the brain are already living in such a high HA-rich environment, when you start tweaking this, this might really cause a disbalance that's really detrimental to those type of vessels. And thus, does this then lead to the formation of vascular lesions there, whilst it does not in the rest of our body? So to study this, we moved to our 3D model of human microvessels. And really what this is it's a tiny device. It's about the size of a $1 coin, where we can pour a little gel that mimics that has the contents of, for instance, the brain in that gel. And we can let that gel set around an acupuncture needle. And when you pull out that needle, really what you're left with is a tube inside an environment that you're interested in. And then we're seeding that tube with those endothelial cells. And then what you get is this really nice, as I showed previously, this really nice 3D blood vessel in this environment that we think is physiologically interesting. And also this vessel is on the blood flow, because we can connect that to media. It's fully connected to media ports, and we put those devices on a rocker. So it's experiencing this movement. So also the cells are experiencing flow more similar to what they normally would in our bodies. So to model CCM disease, the first thing that we did is knock out CCM1. So this is one of those genes that we know causes the disease in familial patients. And when we knock out that gene, what we found is really the hallmarks of the disease that we're very familiar with. So we see that the cells are much larger, and also they express much less of this V-cadherin and beta-catenin proteins here, which really are those cell-cell adhesion proteins that keep the cells together. So that really shows nicely that when you don't have CCM, the cells don't adhere very well to one another. And obviously, again, that would lead to those leaky vessels. So then the next question we wanted to answer was, is the HA content really altered in this disease? And the way we went after this is we went to have a look at all the genes that make the distinct sizes of HA that we know are so important. And we found that when CCM1 is lost, we get an enhanced production of really large size HA, And these also are not fully degraded as they would be normally. So you get this disbalance on what we find in the environment of the CCM cells. So we get excess of the large HA and we're missing some of those small fragments. So then would this lead to formation of actual vascular lesions? And to study this, we started to put these cells into our 3D model. And this is just an example of wild type vessels and CCM disease vessels in that system. And in this case, we grew them in our standard matrix. So here we haven't really changed anything to their environment. This is like the basic environment that we always use. And in this case, really, the cells look pretty similar to our 2D culture. So we find larger cells, and again, they have reduced cell-cell adhesion. So really, they behave as expected also in this 3D environment. And then what we then did is we started to supplement that environment with all these different fragments of HA to see whether if we would add either small HA or large HA, how does this change the outcome of the CCM cell? So how does it change the disease phenotypes? And the most interesting really for us was this outcome here, where we supplement the environment with a small version of HA. And as you might remember, that's the one we think might be lacking just based on what the cells are producing. And what we found in this condition is that the CCM cells are not as large, so they're smaller in size, and also they start to express more of those cell-cell adhesion markers. So overall, that would lead to a more kind of healthy vessel, because the cells are not so big and expanded, but they're like nice and small and better connected to one another. In addition to that, and what seems to be even more beneficial environment for these diseased cells is a mixture of the small and large. And we did this by putting them in an environment that has a lot of that large HA. And in addition, we add an enzyme that then chops that up in lots of different fragments of HA. So you get a mixture of small and large in this case. And that almost completely rescues the disease phenotype. So now we find cells that have a normal size and really express much more weak adherence. So here we're thinking we're changing a vessel that's like very fragile and leaky into a vessel that's now much more stable and would be predictably less leaky as well. So we think that with these studies, really we're starting to understand why CCMs might happen in the brain, and that might be because the brain is so rich of HA, and when you start to then modify that HA, that effect will be even enhanced in that kind of environment. And really this also, we hope this knowledge really helps further thinking about this disease, and maybe what we should do is to try and modify the environment of the vasculature instead of the vasculature itself to try and prevent these big lesions from happening in patients. So this is really where we want to take this work further. So with that, I would like to acknowledge my team here at the top. Christopher Chen in Boston has been tremendously helpful in setting up this 3D microvessel system. Alan Rowan helped us with making the synthetic gels that we use to mimic the surrounding. Emma Gordon, who you just heard spoke, we collaborate a lot together, and we have a lot of vascular fun, as I put it here. And obviously microscopy and the zebrafish facility, because we rely on them a lot for all of our work. And all of you for your attention. Thank you.
Thank you so much for that, Anna. That was just absolutely incredible. I think one of the things that strikes me from both yours and Emma's presentation is just how some of the most devastating things that can happen to us as humans are visually beautiful. So you can see between both of your presentations, you've got a lot of the imagery from the microscopy that it's really quite inspiring and stunning to look at. So I don't know what that's the seed of in terms of what you can investigate further. But I was a bit flippant earlier when I referred to the extent of big tech and incredible equipment that we can tap into here at the Institute. And the IMB microscopy facility alone has the better part of $10 million plus of investment from the Australian Cancer Research Foundation and other contributors. So it does take quite a lot to enable and empower the quality of research that we're sharing today. And we're delighted to have you with us, Anna, to do this. Thank you so much. So last but by no means least, I'm going to hand the microphone duties across next to our third panelist. And so Nat Saez, Dr. Nat Saez, she's actually a toxinology researcher. So we come at it again from another angle again, and she's really passionate about the discovery and characterization of new drug solutions, but with an edge. So being a member of the King Lab here at the IMB means that Nat taps into one of the largest venom libraries on the planet. So she looks to venomous arthropods as a source for potential new drug solutions that can change lives for the better. And Nat has been absolutely integral in being able to scale up the extent of the HI1A molecule that was identified by Glenn and is now working in partnership with Nathan Palpant to develop solutions to protect the brain in the event of an ischemic attack from a stroke, an ischemic heart attack, and even looking at ways that they can make hearts more viable during transport to get to the recipients of those much needed hearts. And so with that, I'm delighted to hand both control of the mic and the slides over to Nat and have her share her incredible contribution to today's theme.
So today I'm really excited to tell you about some research we've been doing here at IMB involving HI1A, a heart-saving drug candidate from a deadly Australian funnel web spider. So in the King group, our interest is really in disorders of the human nervous system where ion channels are involved. And you can think of ion channels as doorways that open or close in response to sensing a particular stimuli, in this case acid, and they send a signal to the central nervous system. Invertebrate venoms are rich in peptides that can modulate these ion channels. They act like keys that can lock or unlock the doors, and we can use them therapeutically either to increase helpful signals or to decrease bad signals. And venoms from creatures such as spiders, scorpions, and caterpillars actually contain thousands of different keys that can modulate thousands of different ion channels. And like locks, we need the right key for the right door. So one of these really exciting keys that was identified in the King group by Sandy and characterized by Irene is called HI1A. HI1A is identified from the Fraser Island or Gurry funnel web spider. In this center panel, we can see the functioning of ASIC1A under normal conditions in the black line. We can see that the channel opens and sends a signal in response to an acidic stimuli. When we start applying increasing concentrations of HI1A, which you can see here in the red and green lines, the channel and the amount of signal that gets through the channel slowly starts to decrease. And HI1A is quite interesting to us in comparison to stereotypical spider peptides because of its large size and complex folding. There are a few properties that make HI1A quite ideal as a drug candidate. Firstly, it has really high selectivity for the ASIC1A subtypes over the other subtypes of ASIC. And even at high concentrations, it has over 2,000-fold selectivity for ASIC1A over the other ASIC subtypes. So this is really promising in terms of minimizing side effects of the drug. The other really exciting thing about HI1A is it seems to have a long-lasting effect. Compared to stereotypical spider peptides like PCTX1, whose effect is really gone after about 10 to 15 minutes. For HI1A, even 30 minutes after application, we're still seeing up to 60% inhibition. So we know that stroke causes the brain to become acidic, and we know that ASIC1A is implicated in neurological damage during stroke. So we wondered whether we potentially had a stroke treatment on our hands. So stroke is the second leading cause of death worldwide. If you're lucky enough to survive a stroke, chances are you'll be left with some serious long-term disability. And it actually accounts for around 3% of global healthcare expenditure. As Anna briefly mentioned, hemorrhagic stroke is sort of in the lower minority of strokes, caused in around 15% of cases, and it's caused by uncontrollable bleeding, and there really aren't any surgical therapies available. The other 85% or so of strokes are called ischemic strokes, and these are caused by a blood clot in the brain. There's actually only one drug called TPA for ischemic stroke, and it's only, sadly, used in less than 5% of stroke patients. A, because it has a narrow window of administration, it can only be given up to 4 hours after a stroke, but more importantly is that it actually increases the risk of inducing intracranial hemorrhage. Now, this would be really, really bad if given to a hemorrhagic stroke patient. So ultimately, this means that a patient has to be diagnosed at a hospital before any treatment can be given. So talking about ischemic strokes, where the cause of the stroke is actually a blood clot in the brain, which reduces blood flow or completely stops blood flow, and that leads to a lack of oxygen in the region of the brain. And the region local to this clot is called the ischemic core, while the region a bit more distal, but still affected by the lack of oxygenation, is called the peri-infarct zone. And traditionally speaking, it's thought that the ischemic core is absolutely resistant to any sort of treatment. So we wanted to give HO1A, a low dose of HO1A, to check it in a preclinical model of ischemic stroke. And what we found actually is, just to explain this graph a little bit, here we have the infarct volume, which is the actual size of the stroke, and here we're looking at the peri-infarct zone. So when HO1A is given 2 hours after a stroke, we see that the infarct volume is decreased by 83%. When we give HO1A 4 hours after a stroke, we can see that the infarct size is actually reduced by 89%. And even when we give HO1A up to 8 hours after a stroke, we actually see a 65% reduction in that peri-infarct zone size. Furthermore, and what was really exciting, is that we actually were able to show that there was some improvement in the infarct volume, even in that ischemic core, which is thought to be extremely resistant to any sort of therapy. And this, of course, you can see in histological slices where we can see that those that were given no treatment actually have a much larger stroke-affected region than those treated with HO1A. So because HO1A has the benefit of not inducing any sort of bleeding, we think it has a really high potential for use and delivery by first responders. So this is, we're talking here about ambulance people. It would really mean that using HO1A means that patients don't need to get to the hospital first. So we can really start treating that potential brain damage at the earliest possible time point. We also have a long treatment window. So far, we've only checked up to 8 hours after stroke, but it means that we have a larger cohort of treatable patients. And this is going to be particularly important for those in regional areas that actually have a really long distance to travel to hospital. And furthermore, it's actually compatible with current reperfusion strategies, such as those where they mechanically go in and remove the clot. And all of this research is an ongoing collaboration with Claudia McCarthy and Rob Widdop from Monash and Lachlan Rash here at the University of Queensland. But we started to wonder if HO1A could actually help after other ischemic events. The problem was, though, that HO1A production was actually really challenging. Compared to a standard spider peptide, PCTX1, HO1A is only twice the size and only has twice the amount of disulfide bonds. And yet the number of potential folding patterns that these disulfides can form in actually leads to around 10,000 different individual folds that this peptide can possibly fold into, but only one of these is actually the correct active fold. And so this really impacted on our expression yields. And we were finding that we're only getting around 100th the yield of HI1A compared to other standard peptides. So this is really where I came into things around looking at the optimization of production of HI1A. And so this is a very brief summary of all the types of things that were involved, and I've only listed those that were actually very integral to the improvement of the production. But all the optimization really lay around the media that we were using for growth, aeration of the cultures, the different growth and induction temperatures and duration, as well as tag cleaving conditions, and even down to simple things like the type of plastic ware we were using. So in the optimized, once I was able to optimize the culture conditions and expression conditions, we're actually able to get up to a yield of 1.5 milligrams per litre. If we were to have used the old expression conditions, this means that I can actually, what would have taken 30 litres to produce, I can now produce in 1 litre. So with a fairly reliable medium yield production method, we were actually able to continue with some of our other exciting experiments. So the next thing we wanted to look at was actually different organs in the body that are susceptible to ischemic injury, so lack of oxygenation. And what we saw was that the heart was highly susceptible, and that's not too surprising really, given its large requirement for energy and oxygen. Now, I will just say before I continue that we do have to use some animal models in our advanced stages of research, but we do make all attempts to only use the minimum number required for statistical significance in robust assays, and when all alternatives to animal models are exhausted or inadequate, or where it's a regulatory requirement for development of HI1A. So here we have an animal heart on a rig, and it is receiving a flow of oxygenated solution that mimics blood. We can actually induce ischemia by stopping that flow, and we can observe that by a complete drop in the pressure in the left ventricle of the heart. When we can actually restore the blood flow, this mimics a situation called reperfusion. And we can see that under these conditions, that hearts treated with HI1A actually recover significantly better than those with no treatment. Now, the majority of this work has been performed by Meredith Red, and she actually gave a speech at last year's Meet the Researchers on the involvement of HI1A in heart attack and cardiac disease. So today I'd like to tell you about a slightly different aspect, which is HI1A in heart transplant. So heart transplant is actually the most effective therapy for end-stage heart failure. And there's around 30 million people worldwide living with heart failure. And it costs around $2.7 billion per year. Unfortunately, what most people don't realise is that in Australia, 70% of donor hearts that are intended for transplant never actually get transplanted. And there's a few reasons for this. It's to do with the duration of storage of the hearts. At the moment, we can only store them for about 4 hours. If the hearts are too damaged to be received by a recipient, or if they have too far to travel. So, for example, if someone in Perth would like to donate a heart, but someone in Sydney needs a heart, by the time the heart is actually transported, it's already over the 4 hours that it can be stored. So, unfortunately, it's a bit of a shocking statistic, but the donor heart demand, there's around 25 heart failure patients needing a heart for every heart that's available. So a potential solution would to actually be to increase the number and quality of donor hearts for transplant. And we wondered if we could achieve this by using HI1A. So this is some very exciting research going on with our collaborators, Peter McDonald and Sarah Scheuer through the Victor Chang Cardiac Research Institute. So this is probably, again, quite surprising to most people, but the way that a heart is transported in most cases for transplantation is literally in a plastic clip seal bag in some solution in an esky on ice. And this is the type of situation where we have a brain dead donor. And it's a pretty standard storage method. Normally, the hearts using this method can only be stored for 4 hours. So in this situation, they've really, where we wanted to test HI1A, they've really extended that out and doubled the time up to 8 hours. And what they've found is that using 2 measures of cardiac effectiveness and measurements of the heart in terms of aortic flow and cardiac output, we can really see that those that were treated with the HI1A recover much better than those that were untreated. In fact, the untreated hearts here are so unhealthy that they would not be suitable for transplant, whereas those treated with HI1A actually have a decent chance. So the next thing I want to tell you about is actually super exciting, and it's really being pioneered by Peter McDonald. And this is donation after circulatory death. So this is another way of potentially increasing that donor heart pool. As I mentioned previously, the current sort of major strategy for transplantation is actually to take it from a brain dead donor. So the difference here is that the heart's actually in asystole. It means it's not beating whatsoever. There's a complete absence of any sort of contraction. And this is the most serious form of cardiac arrest, and it's usually thought to be irreversible. So here we have some videos of actually the process of donation after circulatory death. So what we're talking about here is a patient that is on life support, that is having their life support withdrawn. And this is essentially what actually happens to the heart during this time period. So we call this a period of warm ischemic time. This is obviously in complete contrast to those hearts that are being stored in an esky on ice. And you can imagine that during this warm ischemic time, damage to the heart is constant and ongoing. And so once these hearts actually stop, we're in asystole here, where it's coming towards asystole, where there's no more contractions. There is a standoff period, of course, to make sure that this, after withdrawal of the life support, that the patient is not going to come back. And it's after this whole sort of time period and standoff of period that the heart is actually extracted and then preserved for transplantation. So we have, or Peter, I should say, has found that HI1A actually helps to recover hearts by donation after circulatory death. What I'll show you here is, in the light grey, is that hearts that receive a sort of, that just stay in a standard storage solution really don't do very well. They don't come back. Whereas compared to this solution that we show in black, is actually kind of the best case scenario solution that Peter has found. Although there is one extreme downside to it, which is that this ingredient, zanipride, is actually quite dangerous, and we can't give it to live patients, or it can't see a human type of thing, really. What we were really amazed to find is that actually HI1A was able to compete with this sort of best case scenario solution that Peter had found. And what I'm really excited to show you is that when we removed the zanipride from this solution, take the base solution and combine it with HO1A, it actually completely outperforms any previous sort of best case scenario. So the take-home messages and future directions are that ASIC1A is intimately involved in ischemic injuries of the heart and brain. It has the potential to benefit stroke, heart attack, and heart transplant patients. And my future directions are to make more HO1A, while the method that I have come up with has been able to sort of just keep us going in terms of being able to test all these different applications for their next more advanced stages of clinical testing, preclinical testing and clinical testing. We definitely do need another method. So we're looking at synthetic alternatives there. There is preclinical testing ongoing in heart transplant and heart attack. And in stroke, we're also exploring dosing and route of administration. Sadly, HO1A does not cross the blood-brain barrier, but we do have a student looking into adaptations that we can explore to hopefully get it across into the brain. And just a few acknowledgements, if I could. I have said we a lot in the presentation today, but really the main players involved in all these collaborations and collaborative experiments are these guys here on the left. I've also highlighted some people from the King group that have really been main players in this research in purple. And of course, many thanks to Glenn King and our funding bodies. Can't forget the spider. And a special shout out to Eleanor, who has been devoting a lot of time to helping me make HO1A that we need for these studies recently. And thank you very much for listening.
The Q&A, and I can see that we've got one popped up in the chat for you already, Nat, so that's fantastic. But by virtue of having the power of the microphone to myself, I get to ask the first question. And I think in honor of us bringing together this collective of inspiring, dedicated, truly curious females around the idea that Valentine's Day is around the corner, I'd love to know from each of you, what are you actually passionate about in your job? What do you love about your job? And we might start with Emma on this one and go back, then Anna, and then Nat.
Sure. I would say for me, what I really love about this job is being able to kind of see what happens as it happens, which enables us to really kind of understand how disease occurs. So you probably noticed, like from my talk and from Anna's talk, there's a lot of kind of visualization, and we really love to kind of watch things happen as they happen to help us understand things. So that for me, it's really the kind of visual aspect and the watching of things happen that really drives me in my job.
Absolutely. Definitely very visually inspired, everything that you do, Emma. It's some of the most beautiful stuff that we get to come across. Anna, what gets you up in the morning to do science?
You know what? Obviously, Emma took some of my thunder here because I love live imaging. But I would add to that what I love is, in addition to that, training the next generation. So I really like having students come in, take ownership of their projects and kind of together finding out all these novel things that we didn't know before. So I think that's also something that's really exciting to see happening in front of you.
Fantastic. And Nat, I know you alluded in your presentation to, like, it's been a 2-year challenge to really get together the robust quantity that you needed to propel forward the research with Glenn and the team. So you've got to be passionate about science to persevere in the face of that challenge. What are you passionate about with your research?
I mean, I just love a challenge, Kamira. The more challenging, the better. But I mean, it's just mind-blowing that these spiders and you know, things that we consider to be so scary, and that the potential offerings in their venom, I mean, and the ability to go from kind of characterization, finding a brand new molecule and going, oh, this looks interesting, I wonder what this does, and then actually, almost getting to see it out through to sort of a commercialization stage of wow, we think that this is actually going to really save lives. I mean, what more do you need to drive you every day?
Yeah. You've definitely got big stakes, each of you, in terms of the knowledge creation that you're involved with. It's really inspiring. We have 3 tremendous examples of change makers on the panel today. We do have a question that's come in on the Q&A, so I'll remind everybody who has stuck with us to this point of the webinar that just down in the bottom of your screen you're going to find the Q&A spot, so please pop your wonderful questions in there. We'll try and get through a few of those before we need to bring it to a close today. But from Annabelle Bissett, so she wants to know, with the HI1A, if it's approved as a treatment for prevention of ischemic damage, when could we guess that we would see it being used by the medical profession? And it is a bit of a, there's a lot of factors to that, Nat, but I don't know if you want to talk to that question.
Yeah, so it's definitely a big question, so we're at the moment, we're sort of in the stages of preclinical investigation and research. And we're sort of heading to some of the sort of later stages of that preclinical. Glenn, my supervisor and the group leader, has with another group leader at IMB, has actually just founded a company for the commercialization of HI1A. So we're really hoping that will kind of advance things at a bit of a faster rate. HI1A was actually discovered around 2009, so that gives you a bit of an idea as to how long it has been already. A bit of a side note to that is it also depends a little bit on the application. If we, and unfortunately I didn't get to really show you those slides, but we've got brilliant data on HI1A in heart transplant models. And so I think in that sort of a situation where it's actually being able to preserve hearts intended for donation that have been taken from a donor, the drug never actually enters a human. It only sees the heart when it's outside of the body. So in terms of regulatory requirements for actually being able to sort of get that through, it's a little bit more of it's never a simple process, but it's slightly simpler than for a drug that will actually be entering a live human. So in that sort of a situation for heart transplant, that may actually be where it comes into commercial public use first, because of those sort of a bit slightly more relaxed things. But yeah, certainly we're looking at developing it for a few different things, and
really at an exciting time for you guys with
absolutely
what you've been working towards. So I can see that there's more questions popping into our Q&A here. Now, Margaret McCann asks, and I'm not quite sure who she's directing this to, how can you separate the well-folded peptides from the ones that are not folded as required? I'm assuming that might be around your work still, Nat, but if I'm getting that incorrect, please jump in.
No, you're absolutely right, Kamira. It can be a challenge, but generally speaking, the non-active or the different folds that aren't the good one that we're looking for, they tend to be a bit less stable. So there are certain ways we can try and actually crash them out of solution, for example. But we also actually have some quite advanced purification systems at the IMB, where we actually can do sort of all sorts of, we say orthogonal purification, where we employ a few different types of separation techniques based on different properties of the protein, of the peptides. And then, eventually, once we get a few of them lined up in a row, we can actually get down to the point of having one, being able to get that really one fold that we're looking for.
Oh, wonderful. Thank you. I do just want to highlight a lovely comment from Sheena Burnell in the chat. She said, fantastic presentations from these amazing women in STEM. And I could not agree with you more, Sheena. Now, Emma, I have to be really honest. I've realized particularly the last 12 months, that there's two things that really energize people's interests in the work that we do here. And it's become a case of the spider versus the COVID. So I'm quite shocked that we haven't received a few more questions but your way. But I'm really intrigued with the work that you're doing with, as you quite rightly alluded to, a powerhouse collective of female researchers. How does something like that come about? How do you happen upon this concept and then galvanize together as a team to pursue the research as you've shared with us today?
Probably everyone's a bit tired of hearing about COVID, Camara, so maybe that's why I'm getting less questions, which is totally fine. So I would say this project really came about because all of us are quite good friends as well. So we just started talking about it, honestly, over drinks one day. And it really kind of spurred from there that more and more people kind of started to join in. And it kind of snowballed into pretty much a large majority of the women at the IMB working at this project, and not only at the IMB, but also over at the School for Chemistry and Molecular Biology. So I think it's very important that we're all that the IMB is a very kind of collegial atmosphere, and everybody's very friendly with one another and very open to collaborations. So that's really what drives all of us working together, I guess, just kind of sitting outside, relaxing, having a bit of a drink and discussing science. Quite often happens here.
Fantastic. So you were delivering on our endeavour to put the brightest minds, co-locate them together when possible, and surround them with incredible technology and facilities, and let you guys essentially have at solving some of the biggest issues that we're facing on the globe at the moment. So that's really wonderful. Now, one thing I know that's very close to your heart, Anna, absolutely no pun intended there, is through your research, you've really sort of galvanized around CCM and what consumer groups are going through. So there's a question here I'd love to put to you. So for CCM, if it's caused genetically, is it better to genetically fix the mutation or use HA as a treatment for the long term?
Wow, what a great question. I think if we could fix the mutation genetically, that would be, fantastic, because obviously then none of this would happen in the first place. But to be able to do that, I guess that would mean that you would have to fix it even when it's just a fertilized human egg, which those kind of things at the moment are ethically really not possible. Although obviously technologies like CRISPR are starting to fix these kind of genetic diseases, or at least there's research into that. I think for now, whatever we can do to prevent severe outcomes would be great for the patients that we have. And obviously these kind of studies are running in parallel. So I think, yes, changing it genetically would be fantastic. But in addition to that, trying to avoid severe disease would be also very good for those patients suffering.
And you did point out earlier that I believe it's the 28th of this month is the Rare Disease Day, and it's a chance to spotlight on that. IMB has a strong history in contributing to combating rare disease, particularly through a genetic approach. But are you aware across the scientific community of anybody who's taking a genetic approach to CCM?
I mean, genetic approaches as in trying to identify novel genes that cause the disease. Yeah, definitely. Also, there's a lot of knowledge out there now about which genes are dysregulated in the disease. So it's not only CCM1, but kind of after CCM1, there's a lot of other genes that get dysregulated. So there's a lot of studies into that as well to see whether if we can tackle one of those genes early on, then maybe that can also fix what's happening. So definitely there's a lot of research into genetic changes.
So in my non-PhD mind, I'm hearing that it's an incredibly long road to that kind of solution, which makes the work that you're doing so much more fundamental in the short term for creating solutions for people in the community that are faced with this.
Yeah, and as always, I think all these different aspects of the disease and people are studying different processes. I think in the end that all kind of contributes and comes together, right? So I think we need to know the basic knowledge that from all these different angles, and then hopefully together from that you get the best cure, right?
Yeah, absolutely. I believe we have a question from Kim, but it might be further back up the chat box. So Kim, can I get you to pop that into the Q&A box for us? That'd be really fabulous, because that would make it easier to find. And it would be really remiss, given that we've highlighted what a special day today actually is in regards to females and young girls in the world of science. So there's a reason this day is happening, right? With something like only 30% of the field are actually female, and you certainly need that diversity of think and diversity of approach to tackle and overcome big, hairy goals. So it's really important that we do shine a spotlight on this and let young women, young girls know that this is a really dynamic, exciting pathway. They get the chance to connect and meet incredible women like yourselves. But there are inherent challenges for females in a pathway in science, be it in academia or in industry. So from each of you, what is the one thing that could address this barrier, and what would you like to see happen? Emma, are you brave enough to go first?
This is a very difficult question, Kamara. I'm not sure if anyone has an answer to this. I think, honestly, just with us giving presentations like this and actually providing exposure as female scientists to the younger generation, to show that, you know, it can be done, and there are certainly female role models out there, and not only us, people certainly above us. I have a huge range of female role models. So I think that level of exposure is important, and really to just, you know, keep encouraging women to come into science and make sure that they don't, I guess, maybe get pushed out at a certain level because they maybe want to have other interests. So that kind of thing, just kind of making the workplace inclusive and flexible for women, and exposure, I would say.
Thanks, Emma. Nat, would you say there's something that you've had access to or has helped in terms of your trajectory in science?
That's a really good question. I guess as a young undergrad student, I had the opportunity to enter the advanced studies program in science, and that actually, out of that, through my interactions with researchers that we heard presentations from and things like that, I'd go and talk to them, and a lot of the time I'd found that they were actually just really happy to talk to you know, young inquisitive minds. And even I actually was offered quite a bit of sort of lab work experience even as an undergraduate in these research labs at UQ, and that really just got my passion locked in right from an early age. And I went to manage a pharmacy for a couple of years in between my undergrad degree and my honours and PhD. And I had just missed being in the lab. I really did. So I came back, made a beeline. I think it helps that one of my first supervisors during that undergrad experience, and I came back to her lab for honours, was a female group leader. So I did have that kind of role model from the get-go. And I think, yeah, luckily I've been in labs such as Glenn's that have just been really supportive. And yeah, I don't think actually, and if I could just add one last thing to Emma's answer about with the previous question, I think one of the things that one of the things that some of the funding bodies are doing as of late is actually de-identification on grants. So you can't tell the person's name that's applying for the grant. You can't tell whether they're male or female. And I think that will actually go a long way as well to seeing that females are a bit more recognised, perhaps.
Fantastic. Yeah. And so, caveat before I hand over to Anna, she is actually the chair of our diversity and equity committee here. So this is an issue that she's got a real passion and insight around. So Anna, what would you like to see?
I think Natalie touched on it really well at the end there. And I think representation obviously is one thing, so it's very important. But progression, I think, is the biggest hurdle, where we see that big drop off of female representation in science. And a lot of this, we're now starting to understand, comes down to that bias. So there's conscious bias towards females, but also unconscious bias towards females. And yeah, so having that change in the granting system is great, but also trying to train people in these different forms of bias and to make them aware of their own unconscious bias, to try and see whether then, I guess, across the board, everyone can be judged equally. And that would hopefully help women progress through those difficult stages.
Fantastic. Thank you so much. Listen, I'm very cognizant of time, and I'm grateful for those of you who are still with us online. There are a couple of questions there, so we will get any relevant questions through to our researchers who are online today and follow up with you after the fact. But I do need to close out with just a couple of formalities. So we do the Meet the Researchers. This is the first of 4 that will be happening this year. So please, we'd love to get some feedback from you. It's really important that we take information out to the wider community and frame that in a way and offer that to you in a way that has some meaning to you. So I do invite each of you to offer feedback through the QR code that's on your screen at the moment. And importantly, this will give you an opportunity to sign up to our e-news or look at any of our publications if you want to, so you can learn more about the incredible work happening at the Institute. Gives you a chance to stay in touch with us and learn about events in the future. Also, it'll help you in the one challenge we want you to take away from today, which is to spread the word about this incredible collective of research and the ambition to make the world a better place at large. Another way that you can actually make a critical contribution to the work that's being pursued here at the Institute, and I do want to just take a nod to several people who are online with us today. We're delighted to be joined by Selwyn, Alan and Wendy Grummet, and Pauline North, who are all dedicated members of our Directors Circle. They're also joined online by people who've given to the Stroke Research, our Women in Science and Technology Fund in the past, and also our Ignite Innovation Fund. So our Directors Circle is a collective of like-minded individuals and companies who share in our vision to make a better world through incredible discovery that you've heard about here firsthand. They have access to VIP initiatives. They spend some time with the director or some of our senior research team here. They come along to special events and initiatives and ways to stay connected to each other as a cohort almost of people who have a genuine passion to help us create change. So if you'd be interested in joining the Directors Circle, certainly go online and learn more and find out how you can make an impact with us through philanthropic contribution as well. So as I mentioned, this is the first of 4 Meet the Researchers. The next one we have is actually happening on International Motor Neuron Disease Day. So we're delighted to bring to you 3 of our incredible genomics experts who can share with you exactly what they're doing in this space and how they're contributing to what is just a horrendous issue for those that are more familiar with ALS or MND. And then in September, we're going to touch on what we're doing in the chronic space. So more information will be coming to you shortly. And then we've kicked off in the last couple of years with our PhD candidates that are coming through. They've really galvanized their energies around critical issues such as inflammation, chronic disease, infectious disease as well. So we'll have the opportunity to revisit at the end of the year how our global challenges scholars are actually making a difference through the work in their early career research efforts. So please stick with us across the year and be involved in these valuable conversations. And still in the theme of incredible, inspiring women, we're delighted to share with you there's a UQ event on the 9th of March in recognition of International Women's Day, and it brings together 3 inspiring women who have connections to UQ in different ways that are really, they're forces for change in either health, technological development, or medicine. So we love this event because hearing from these remarkable women will certainly give us the opportunity to learn, be inspired, even the next generation of female researchers as well. All funds raised from this event will be dedicated to the work that we're undertaking here at the Institute into ending endometriosis, which we alluded to before. It's a genetic approach, so it is the long game. But if you'd like to learn more about what we're doing or be involved in that breakfast, please go online, book your tickets, and we'd love to see you at Customs House on the 9th of March. That brings us to a close for today. I would just like to end by saying thank you so much for your interest and your investment of time today and coming and learning more firsthand. And I'd also like to say a huge thank you to Emma, Anna, and Nat for your incredible volunteerism of time today. I think it's really evident your time is important and there's things you need to be doing. So coming and helping us share the word today is absolutely valuable, and we're very grateful. With that, thanks for joining us, and I'll say goodbye now. Please be safe and stay connected to everybody out there in the community. Good day.